Welcome to yet another Zoom call organized by Jungle Bells. Jungle Bells is an exclusive women's wildlife group wherein into wildlife related activities, conservation, and wildlife tours. Before we begin, I would like to request all of you to turn your cameras off and switch off your microphones, please. If you have any questions for the guests, please type them in the chat box and we will address them after her talk. Also, if you want to receive any further communication from us about our events, please type in your email addresses and phone numbers in the chat box. To introduce the speaker of today, Pragati Abhyankar, I will hand over to Aarti.
Thank you, Hemangi. So a little bit about our guest for today. Dr. Pragati Abhyankar is working as an associate professor in the Department of Microbiology, Haribhai V. Desai College, Pune. She has done her graduation and post-graduation in microbiology from Abhasaheb Garware College, Pune. She completed her PhD from SPPU. She has a teaching experience of over 28 years in microbiology. Pioneer in setting up the Department of Haribhai V. Desai College, she has nurtured her passion for teaching the subject. She has published research papers in national and international journals and completed research projects. Her main field of interest are biochemistry and industrial microbiology. Wow, Pragati, that's really a very illustrious career, education-wise. We look forward to hearing more from you on this subject. So over to you.
Thank you, Aarti. Thank you so much. Good morning, all of you. It's nice to see so many different people here. Since years, I have been teaching microbiology, and it has really been an interesting journey all through. So today I'll be sharing a few very basic facts about microbiology, microorganisms rather, whom we have really see the unseen world, in fact, which exists all around us. The photographs, the journey of Jungle Bells, which I saw just now, shows you so many things which you can see with your eyes, which is in front of you. But this world of mine, the microorganisms, is something which exists everywhere around us, but unfortunately we cannot see them, only feel them. And especially last 1.5 years, we have been so much with it. And since the COVID-19 came all around us and suddenly hit us like anything. And when we all got used to that word virus so much, those times also we did not see the virus, we could not see the virus, but we felt it. And then suddenly we thought that, yes, this is important too. So I'll just take you through an interesting journey, trying to make it interesting for all of you. For a journey which started long back, and then today we are here with the present status of microorganisms. So give me a minute to share my screen and then we'll go through it. If you have anything to ask, anything to share with me, let us do it at the end of my presentation, so that we are completely through what I want to show you, and then I can listen to what you want to know from me. Okay, so just give me a minute, I'll share my screen and then we can move forward. Yeah, can you see the screen?
Yes,
Yes, but We are unable to see the presentation yet. Yes, now we can see the presentation.
Okay. Yeah. You can see the presentation mode now?
Yes, yes, it's visible now.
Okay. Yeah. So my presentation says it's an amazing world of microorganisms. Of course, not seen by the naked eyes as such. So we'll start with the slight terminology that we normally use in this subject, that is microbiology. Students, especially after their 8, 9 standards, are very much fascinated by this word microbiology, which is indeed interesting. But what it is study of microorganisms, or as popularly called as the microbes, a diverse group of minute, simple life forms that include bacteria, archaea, algae, fungi, protozoa, and viruses. Of course, we are more well versed with these two terms, bacteria and viruses, which we commonly think are the only microbial forms that exist. But no, under these microbes, we also have archaebacteria, we have algae, we have fungi, we have protozoa. All those which we cannot really see with naked eyes are included under this study, or under this term, what we call as microorganisms. Way back in 13th century, these living invisible entities were felt. It was realized that something which we cannot see is responsible to cause decay and to cause disease. In 17th century, these living forms were first discovered. In early 19th century, the word microbe was coined for these organisms. The science of microbiology eventually developed into a specialized science, into a different branch of life science. Till then, probably botany, zoology was what we were dealing with. But then when it was realized that microbes are there, microbes do form a huge world by themselves, microbiology was developed as a specialized branch of life science. Microbes are a very large group of extremely diverse organisms. So the diversity that we see among these organisms is huge, huge. These are a few photographs or a few types of microorganisms. The shapes, sizes extend beyond these. So you can have rounds, you can have skinny, curved shape organisms, you can have organisms which have tails, or what we call as flagella, which are their locomotory organs. Some look like some give you a flowery appearance, some have shells, some have amoeboid appearance, others look like spaceships, some look like beaded necklaces. So depending upon the type, the genus of the organism, you can have different shapes for the organisms. Now, what are microbes? They are our friends, they are our foes, though we understand microorganisms more to be dangerous because we are familiar with them because of the diseases they cause, the infections they cause, the curable ones, the uncurable ones. So, but it's not that. Microbes are in our lives as our friends, and of course, as our enemies or foes also. Microorganisms provide ample evidence of their presence, sometimes unfavorably and sometimes favorably. We have all experienced it in Corona times, their unfavorable existence, their unfavorable presence all around us. But these organisms are of infinite value to Earth's ecology. Perhaps human race, our atmosphere, our environment will not be the same if microorganisms were not there. Now, today we have studied microorganisms, a variety of microorganisms in such details As I mentioned before, we cannot see them with our naked eyes. Invention of microscope and further development technologically was very important to study the microorganisms. Microbiology essentially began with the development of the microscope. Maybe people had tried to see organisms before that, but it was Leeuwenhoek, Antony von Leeuwenhoek, who was the first to provide proper documentation of his observations. He was a self-taught man in science and is commonly known as father of microbiology, because let's call him the pioneer, since he invented the microscope first, with which he revealed the entire world of microorganism. Antony von Leeuwenhoek is best known for his pioneering work in microscopy and for his contributions towards the establishment of microbiology as a scientific discipline. These are some photographs of microscopes, perhaps none of these ones, the ones which are shown here, are familiar to us. What we see today is a simple dissecting microscope like this in botany and zoology laboratories. And this is the one which is the technologically improved microscope, which we now use today for observing various different kinds of microorganisms. But the pioneer to take us today till this stage was Antony von Leeuwenhoek. The cell. We all are familiar with animal cell, plant cell, a microorganism. Each single organism is a single cell in itself. So this single cell was first discovered and named by Robert Hooke in 1665. There were two philosophies at that time, abiogenesis and biogenesis. Abiogenesis was also called as the spontaneous generation hypothesis, which actually said that life can emerge from non-living things, like decaying meat, decaying organic matter could give rise to life. This was a philosophical hypothesis on one side, which had originated around the time of Aristotle. And it was widely accepted that time, because now also we can actually observe that if you have decaying organic matter, naturally you can see flies, mosquitoes around that. So that doesn't mean that the flies originate from the decaying matter. It essentially is the flies getting attracted to the decaying matter. But then, because this was the observation at that time also, they put forth this theory of abiogenesis, or the spontaneous generation hypothesis. But then later, a scientist, Francisco Redi, experimented widely and put forth the theory that life is necessary to produce life. Life cannot originate, life cannot come out from non-living or decaying matter. So that was a milestone in the journey of microbiology, or in the journey of life as such. This name, Louis Pasteur, is extremely familiar to all of us. We are all very, very familiar with the term pasteurization. All our milk bags, all our milk products, which are packed, now put in Tetra Paks, carry this name: pasteurized milk, pasteurized butter, pasteurized yogurt. So this is a process, the process of pasteurization, which is named after the person who put forth this process, Louis Pasteur, who was a renowned microbiologist, a French biologist, who has immense contribution in this field. Now, this process of pasteurization, which is basically a process to preserve these milk products, so that the microorganisms which may cause decay or which may cause deterioration of the milk products are killed using this specific process. But at the same time, the product should not be harmed. The product should not be altered in any way. That is, after going through this process of pasteurization, the milk, butter, the yogurt, the curd should not get altered with its taste, with its appearance. And this process was first put forth by Louis Pasteur way back in 19th century. So in those times, this was a genuine idea that if you pass the products through this process, they would remain in the same state till they are not exposed to organisms. And hence, after the name of the person who first gave us this process, Louis Pasteur, this process is called as pasteurization, wherein we heat the product at a certain temperature, hold it at that certain temperature, and then we preserve that product with the observation that if done properly, the microorganisms are killed and no further decay of those products will be there. This person, Louis Pasteur, has also to his credit principles of vaccination, microbial fermentation, along with pasteurization. Importance of vaccine we all have realized, but he had put forth the principle of vaccination. We have a large number of fermented products now. A simple example is curd, that is a fermented product that comes from milk to wine, beer, and your organic acids, vitamins, antibiotics, steroids, all in between, which are fermentation products, which are formed due to action of microorganisms, though we never see them. When you are fermenting milk to make curd, you add previous curd, which we call as inoculum. That is, you are introducing microorganisms in that milk, which would ferment that milk, change that milk into a product which is now curd. So we have to give conditions which are favorable to those organisms. That can be temperature of the milk which you maintain. That can be the amount of organisms which you are adding to the to your milk, so that it gets converted into curd. He is remembered for his remarkable breakthroughs in the causes and prevention of diseases, and his discoveries have saved many lives ever since. He reduced mortality from puerperal fever and created the first vaccines for rabies and anthrax. These are both animal-borne diseases. Rabies, we are well versed with in dogs, especially. Anthrax is also a common disease, a common infection, bacterial infection that is seen in sheep, though we don't realize its importance here, but probably in the areas where sheep are important, anthrax also becomes important. This is just to recognize the contributions of Sir Louis Pasteur. After Pasteur, we move on to Sir Robert Koch, Heinrich Hermann Robert Koch, who was a German physician and microbiologist. He is one of the main founders of modern bacteriology. He identified the specific causative agents of tuberculosis, cholera, and anthrax. Now, in those times when the concept of microorganisms was new, Sir Robert Koch isolated identified which organisms cause tuberculosis, which are the organisms that cause cholera and anthrax as well. Today we know everything about these diseases. They are a common word now: tuberculosis, cholera. We have vaccines against them, but identifying it for the first time was indeed a creditable thing. He gave experimental support for the concept of infectious diseases, which included experiments on humans and other animals. He is actually recognized for putting forth what we call in microbiology as the Koch's postulates, in which he has described how a disease is developed and how, if that disease is caused due to a microorganism, how those microorganisms should be isolated from the diseased patient, or the diseased from the sample of the infected patient. And then when you isolate that organisms from a appropriate sample, say can be blood, can be sputum, can be urine sample, can be fecal sample. So if you isolate that organism from that sample, that isolate should again cause the same disease in a healthy individual. So that is the path to establish the causative agent and progress of a disease. So that was first done by Robert Koch, and he is still known for his Koch's postulates. For his research on tuberculosis, Koch received the Nobel Prize in Physiology or Medicine in 1905, and the Robert Koch Institute is named in his honor. So the discovery of the organism for tuberculosis was done way back in 1882, which was a leading cause of death in the world that times. Because the organism was not known, naturally the treatment against that organism was not established. So tuberculosis could not be treated in those times. His work was definitely creditable, that identifying the organism for tuberculosis, studying the organism for tuberculosis, and giving the world a definite direction for treatment of tuberculosis. Now, Ferdinand Kohn, who is considered to be the father of modern bacteriology, his studies laid the foundation for the classification of microbes. Now, if you know plants, if you know animals, you can see them, you can classify them that this is a shrub, this is a herb, this is a tree. So there are certain criteria which we use to classify plants. Similarly, we use certain criteria to classify animals. We need to do this classification because then the study gets a certain direction when you classify a kingdom. So with plants, with animals, it's slightly simpler because you can see them. Like if I am doing classification for plants, there are certain criteria. I'll see the height of a plant, the habitat of a plant, the type of leaves it shows, the type of root system it has, the type of flowers it bears. So all these criteria can be used to classify plants. Similarly, with the animals, the type of animal, the habitat of animal. So these criteria would be used to classify animals. But then for microbes, the difficulty was you cannot see them. So they had to be studied under the microscope. They had to be grown in artificial media. Their colonies were to be observed. Their biochemical characterization is to be done. And then you classify the microorganisms. So this person, Ferdinand Kohn, studied the organisms and he laid the foundation for the classification of these microorganisms. He gave some of the first insights into the incredible complexity and diversity of microbial life. So his credentials are very huge. Today, when we teach microbiology or when we study microbiology, any organism is first put into its proper class after making all the detailed observations on that organism, and then those organisms are studied further. So this, the classification of microbes was an important milestone in the progress of microbiology. Now, after that, till now, with the microscope, the researchers and scientists were only able to see bacteria or maybe algae, fungi, protozoa. But existence of something which could not be categorized into any of these classes of organisms, that is bacteria, algae, protozoa, fungi, but still they existed. They showed some symptoms. They could infect plants, they could infect animals, they could infect human beings, but then they could not be seen with the microscopes which existed till that time. Now we are very familiar with this term virus. It has been of such a common use that from small kids to maybe everybody, we commonly use this term, I had viral infection. I had fever, I had cough and cold, and they said it's a viral infection. And coronavirus made this term so abundantly popular that I don't think there is anybody who doesn't know this term virus. So, but discovery of this virus was much different than discovery of bacteria. Dmitry Ivanovsky, he observed patches on tobacco plants. And from these patches of tobacco plants, he could not isolate bacteria. So naturally, a thought came that this looks like an infection which is caused by some microorganism. But since no bacteria could be isolated from this infection, that should be something different from the bacteria which is causing this infection in plants. Then what he did, he took the sap of the diseased tobacco plant and filtered it with help of bacteria proof filter, so that the bacteria would be retained and the sap would not contain bacteria. Now, if you transferred this sap to a healthy plant, and if the bacteria were removed, it should not show the same disease symptoms in the healthy plant. But that was not the observation. The observation was that this filtered sap, which was free of bacteria, could still cause infection. Now, what was this? So Martinus Beijerinck called the filtered infectious substance a virus. And this discovery is considered to be the beginning of virology. This gave them a thought that these organisms, which could not be retained by a bacteria-proof filter, are smaller than the bacteria, and hence they were called as viruses. Thomas Milton Rivers, an American biologist and virologist, is called as the father of modern virology. as I described the Koch's postulates for progression of a disease put forth by Sir Robert Koch, so did Thomas Milton River put forth the reverse postulate for establishing the infection with the help of viruses. So they started with unseen microbes. They invented the microscope. They observed the bacteria under that, established the relationship between disease and bacteria. And now, as the next step forward, they had established relationship between viruses and infection, the viruses being smaller, much smaller in size than the bacteria. Next, we come to an important concept of antiseptic surgery. We are all aware of surgeries. The environment in operation theater should be of bacteria free, since bacteria can easily cause infections in open wounds, which are easily accessible when a surgery is going on. So post-operative infections were a very big threat at that time, because during surgery, when the skin barrier was opened for performing some surgery inside by breaking open the barrier of the skin. So if proper surgical precautions were not taken, this open skin was a way for bacteria to enter inside. The blood which is present inside a human body is a very good medium for bacteria to grow, to proliferate and cause infection. And so post-surgical risks of infection were very common in those times. And patient was cured of the problem for what he had undergone the surgery, but post-operative bacterial infections proved to be much dangerous, and they were actually ending into end of the life of the patient. So Joseph Lister realized this. He was himself a surgeon, and he put forth the idea of antiseptic surgery, that is, use of chemicals before the surgery to keep away the organisms, so that the organisms won't infect the open wounds and will not cause the dangerous outcomes of the surgery. So this was indeed a great discovery, a great idea put forth by Joseph Lister. He is often called as the father of modern surgery, and the term antiseptic, that is keeping away or doing something to keep away sepsis, that is a progression due to the infection by the organism in the opened skin surgical operations. So this was an important milestone and an important discovery to keep away the organisms. Then we reach another important milestone, which is again a branch of microbiology, which we call as immunology, where we study the immune system of the human body, which basically deals with study of blood in very simple language. Actually, the term immunity has also spread like anything since Corona times. We have seen so many videos, gone through so many WhatsApp messages to boost immunity, to upgrade immunity, right from that medicinal tea to practically anything, vitamin E tablets, importance of vitamin D, importance of vitamin E, importance of vitamin B12, and whatnot. But then the beginning of this concept, or the big study of the immune system of the body, started with Paul Ehrlich, who did a great amount of work in hematology, that is study of blood, immunology, study of immune system, and antimicrobial chemotherapy. Now, this word chemotherapy is also very familiar to us. We relate it to therapy after cancer is diagnosed, malignancy, to suppress the malignant growth. But then chemotherapy basically means the use of targeted chemicals against microbes. Was what the initial idea was, antimicrobial chemotherapy. So Paul Ehrlich first put forth this idea. His idea was that since each organism is different, which causes a different infection, like tuberculosis is different from cholera is different from typhoid. So each of these organisms has some speciality. Maybe it has a specific type of cell wall composition. Maybe it has a specific type of cell membrane composition. It has certain types of proteins, certain types of DNA makeup. So instead of creating a broad antibiotic or a medicine, a therapy could be generated which would kill only the organism which is causing the infection and will not harm the other cells or the other organisms present. So this concept was first put forth by Ehrlich, for which he was given the Nobel Prize in Physiology or Medicine for his contributions to immunology. He put forth the idea that a magic bullet can be created that killed only the organism targeted. Salvarsan was a compound which was created, or which was actually formulated at that time. He identified mast cells, which are a type of cells inside the blood, created the field of differential staining of blood cells, that each type of blood cell, we are all aware of at least three types: the RBCs, red blood cells, the white blood cells, and the platelets in blood. So he put forth differential staining methods for types of WBCs. There are various types of WBCs, the white blood cells. So a differential staining method was put forth by him so that you could observe each of the WBC cells independently. Like I mentioned, you have neutrophils, eosinophils. If you have ever done your hemogram, you get a differential count there, which has these words: what's the count for neutrophils, what's the count for eosinophils, the basophils, lymphocytes, erythrocytes. So the staining technique, so that each type of these cells can be identified separately, because these cells are different, or their nucleus type is different, their shapes are different. So he designed this staining method so that we could observe these type of different blood cells. What I was mentioning is these are the types of white blood cells. Each of them is different. And you actually use that staining technique, the differential staining technique, and you observe these cells, and you can actually count them. You take up 100 cells, stain those cells, you get specified chambers for them so that you can count the cells. And each cell looks different. You can mark the cells. Say you observed 7 neutrophils, you observed 10 eosinophils, you did not see a basophil, you observed a few monocytes, a few lymphocytes. So on basis of that, you can see your report, refer back to your hemograms, which you must have done sometime for many different reasons. And there is a reference range given in front of the count, which is seen in your blood. And then you can match it with your reference range and see if your counts are adequate or not. So this is a routine practice now, but this was initially put forth by Paul Ehrlich, who gave us this idea of differential staining. And now it is this technique that is being used. This is again a different scientist, Mechnikov, Ilya Ilyich Mechnikov. He was a Russian zoologist, best known for his pioneering research again in immunology. He discovered the phagocytes or the macrophages, which make up our defense system, which are a part of our innate immunity, that is immunity by birth, which is called as the innate immunity. These macrophages or phagocytes actually engulf the infectious organisms and kill them. So this becomes a very major defense mechanism in the body. The stronger your innate immunity is, the stronger macrophages you will have, and your defense system will be strong. He also developed a theory that aging is caused by toxic bacteria, because these toxic bacteria liberate toxins in the body, in the gut, which circulates all around the body, causing the cells to age faster, and effectively the process of aging is stimulated. So, and on the other hand, the good bacteria, I'll be talking about the good bacteria also. So these good bacteria secrete various acids, various substances, which help us to prolong good life. Then we talk about Sir Edward Jenner, who for the first time put forth the smallpox vaccine, the idea of how vaccine can be made, because vaccination is actually prevention. It is not the treatment after a disease is caused. It is a precautionary measure taken to avoid disease, avoid infection. We have realized the importance of vaccination with the pandemic. Vaccines have always been with us. As soon as a baby is born, he is vaccinated. We have a specified vaccination schedule. We have vaccines for so many bacterial and viral diseases now, but the first vaccine, which was developed against smallpox, which was a dreaded disease that time, was by Edward Jenner. He's also called as the father of immunology because of such an important discovery. And his work is said to have saved more lives than the work of any other human being. So you can just imagine his contribution. The vaccines which are present there today, which are saving the human life from so many of the dreaded diseases today. The first vaccine was given by Sir Edward Jenner against the smallpox, but he used the cowpox, cowpox as his first site to develop this vaccine. He took the sap from the cowpox and injected it in a milkmaid, and she was saved of this disease. So the idea of vaccination to stimulate antibody production against a specific antigen, that is organism. So that was put forth by Sir Edward Jenner. Jenner's vaccine was the first successful vaccine ever to be developed and remains the only effective preventive treatment for the fatal smallpox disease. So though now is the fact that smallpox has been eradicated, still this becomes the pioneer in vaccination. The beginning of antibiotic era, we are all very, very, very familiar with this word antibiotic, which actually means something against life, something against bacteria. Now the speciality of antibiotic is it's produced by an organism and used against another organism. It's sort of a self-defense mechanism for that organism, but it produces the substance which is able to kill other organism, and we can produce it with the process of fermentation by using that organism and use it. Now this antibiotic is a very, very familiar term. We use it almost against all bacterial diseases. There are antibiotics against all bacterial diseases which kill the organisms, which cure us of that infection. But the first idea or the discovery of this antibiotic, the credit goes to Sir Alexander Fleming, who first developed or first obtained the antibiotic from a fungus, a mold, Penicillium notatum, and that was penicillin. We have a huge penicillin. We had, in fact, should say now, I don't think they are producing it at Hindustan Antibiotics, very close to Pune in Pimpri. We have the huge plant for production of various antibiotics there. And this idea of antibiotic was first put forth by Sir Alexander Fleming. He was again given the Nobel Prize for this great discovery. That was the first antibiotic found out, but now we have a long list of antibiotics which have enabled us to fight against a long list of infections. This is yet another antibiotic, streptomycin, which is also used for many, many different dreaded diseases, infections. And this was another discoverer, a researcher, Waxman. Till now, I think I have talked more about the harmful effects of organisms and of course human beings or humans trying to fight them with their knowledge. The beginning of soil and environmental microbiology comes with Martinus Wilhelm Beijerinck, after whose name the organism is also named, which is the species of called as Beirinkia. So that is the credit of this great person who found out the various organisms which are associated with plants, which are associated with soil, and carrying out different functions due to which they become inseparable from soil and environment. He isolated the Rhizobium species, which is symbiotic with plants. If you have observed the fenugreek or the methi plant, the roots of the methi plant, you can just clean them of the soil, and many a times you observe small nodules which are present on those roots. And if they are absolutely clean, you can see a light pink color to those nodules. And that is because of the presence of these symbiotic organisms, which are called as rhizobium, which form the nodules in symbiotic association with the roots of these plants. And they do an important function of fixing nitrogen, which is required as an essential element by the plants. So important they are. The plants will not survive in the status as they do if these organisms were not there. You have a long list of organisms which do this job of fixation of nitrogen. Many of them are symbiotic, that is, they live with the plants. Many of them are non-symbiotic or free living, as we call them, but they are present in the soil and can be easily isolated from soil. This is a Winogradsky column that we normally make in the laboratory. And you can see different colors here, which are due to pigments of different bacteria which are present in these zones. This is a tall column. It's a glass column, a measuring cylinder or a chromatography column, in which we add soil and waste paper and some decaying matter and water. And slowly with time, as per availability of oxygen, which penetrates through this column of water, you see development of various types of organisms and actually development of color here. This is a very, very interesting phenomena. You can see we actually do this in the laboratory, but this also emphasizes on the useful effects or the requirement of organisms as a part of our ecosystem. Then we move on to the modern era of microbiology, which is a technology-based era. We have evolved various techniques. The familiar to us all now is the RT-PCR, which we have broadly used for detection of the coronavirus. It is a technological or a biotechnological advancement in that field. We are using microorganisms for the production of various foods, beverages. We are studying organisms to keep our foods and beverages fresh and consumable for unlimited times. Now, we have studied the genetic makeup of various organisms. We are trying to play with that so that we can use them for our betterment, for getting good yields of the products that we desire. Beneficial microorganisms such as plant growth promoters, phytopathogen controllers, all this is under study, is being studied, is being used continuously. As I mentioned before, we are using organisms for synthesis of diverse chemicals, fuels, different types of polymers, steroids, antibiotics, vitamins, organic acids, and whatnot, you name it, and microorganisms are used for that. We are using organisms to detect fuel under the ground, many meters under the ground. The organisms are used for that. On the other hand, we are using organisms to clear spillage of petroleum and other petroleum products also. They have huge capacities of decomposition. So we are using that. People are experimenting if you can find organisms which can destroy, which can decompose plastic, which is posing problems for our environment. So these research activities are in full swing now, wherein we can use microorganisms. You can practically you see the presence of organisms in any of these processes. You keep a substrate, and after some time you see it converted into a product. The substrate no longer exists. You cannot see the microorganisms, but you can see the product which has been changed by the microorganisms. So these are technological advancements now. It's going at full pace. That was probably the reason why we could control the pandemic. Now, microbiology, the organisms have diverse applications in field of medicine, in field of pharmaceuticals, in industry, in agriculture, you name it, and you have organisms working there. So you can make them work for your betterment, for getting good results, for using them for combating them itself. So they are going to give you very unique benefits to our society. This is just a flow chart to show wherein microbiology extends itself. The studies can be done under various names: processes, diseases, environment, industry. So they are being diversified into all different branches. The common sources, the microbes. Are microbes dangerous? Not as dangerous as we used to think, because now we have remedies against them. The grand majority of microbes are either not in contact with us, or if they are in contact with us, they are actually mutualistic because they also want something from you. Or many of them are simply neutral. They don't harm you until and unless you fiddle with them. They are dangerous when we make them dangerous. Make them dangerous through antibiotic overuse. Please, now let me have a short look at the probiotics or the good bacteria. The probiotics are also gaining a lot of hold, a lot of research being done on probiotics. As I use the term antibiotic, which means something to kill bios, to kill life. Probiotics is something to improve the life, to make your life good, much better than what it is now. So these are nothing but live bacteria or yeasts that they themselves and their products are very, very good and very, very essential for us. They live in our gut, produce some products which are good for our gut health. And once your gut health is good, your overall health always remains good. Probiotics are often called as the good or helpful bacteria because they help to keep your gut healthy. The good bacteria are found everywhere, all over your human body. And they are in contact with the outer world. They try to combat the incoming dangerous organisms in the body. They are present in the gut, they are present in the mouth, the vagina, the urinary tract, on the skin, in the lungs, everywhere. But their effects are good. They are trying to maintain your good health over and over again. So please don't overuse the antibiotics, which may result in killing of these good bacteria. We need them. There are so many good effects. They help to digest food properly. They keep bad bacteria from entering your body for increasing in number and making you sick. They create few vitamins which our body cannot make. Maybe many a times we consume bad bacteria, infection causing organisms inside, but these good bacteria help to keep them away. They help to break down some medicines, which as a whole may not act that efficiently. They also help in absorption of the medicines inside the gut so that the good effects of the medicines are circulated all over the body. This balancing act is naturally happening in your body all the time. This is a picture of the microbiota that exists in the gut. You have so many benefits of the good bacteria. The good bacteria establish themselves inside our body through very, very simple food items. You, in fact, don't even need to take probiotic supplements, except for the times when the count has really gone down due to some infection or some other therapies. Otherwise, our daily food intakes in form of yogurt, in form of curd, in form of buttermilk, in form of cottage cheese, or some fermented food products, like these are not very similar to us, but in many parts of the world, these are the fermented food products which help to establish the good bacteria. This is just a reminder of the coronavirus, which we have faced with the growing percentage of vaccination, with the growing awareness against this, with the huge amount of research which has been done over coronavirus. We have successfully controlled it, though it hasn't vanished. We are still at a stage where you need to take these precautions because, again, like all those microorganisms, we are unable to see this coronavirus. And so taking precautions is the best way to keep it away. You can successfully keep it away. All these norms have been emphasized from time to time, and what we feel still is you should maintain this till we are sure that yes, the virus is no more with us. We are in with vaccination, which has helped us, is going to help us to put a few steps ahead of the virus. We would successfully keep it away, I'm sure, with this vaccination. But you have to take all precautions to keep it away. Wishing you all good health, all of you. Please take care. We are not totally out of this corona. All researchers all around the globe doing a lot of research to come out of this. Please help them by maintaining your own precautions. Thank you, all of you, for this patient hearing of my talk.
Thank you so much, Dr. Pragati. This was really very interesting and eye-opening. We had studied some of it, very
Yes.
basic, like the history you talked about maybe in school. But
Yes.
after that, you lose touch with these kind of topics unless you're genuinely
Into
studying
it.
them.
Right.
Yes. So yeah, it was very, very enlightening, especially the whole good bacteria thing, which we tend to forget.
Right.
There is always a overuse of antibiotics nowadays, because as soon
True.
as you go to a doctor, they will,
That's
even if
right.
it's a common cold, you get prescribed
Correct.
antibiotics. So that's something we need to avoid as far as possible. So it was a really very enlightening webinar. There's one question from Sanjay Deshpande. He wants to know,
Okay.
is there any connection between these microorganisms and wildlife? Like, do they benefit the wildlife in any way?
Yeah, I mean, I'm not very sure since that part I haven't studied much, but then the general ecosystem in the wild, as we talk about human gut, you do have microorganisms in the animal guts also.
Right.
So maybe the types would be slightly different because the food types are different. So the established microflora that is helping the wild animals to maintain their health, because the microorganisms normally inhabit gut in all ways. Depending on the food you eat, the flora gets established. As a baby is born, the type of microflora changes as the food of the child changes. And then at a certain stage, it gets established and then it performs its function as required. So what I think that animal gut also must be harboring some kind of flora that must be helping the animals to have their health status maintained. And of course, around the animals in the ecosystem, to maintain their ecosystem properly, there is a huge role of microorganisms.
Okay, that's interesting. We never really think about these
Yes.
things. There are three more questions asked by uh, Dr. Samir. He wants to know, recently formation of curd using Hastata house was in news. Is
Yes.
curd formation possible in this manner?
Actually, to tell you the truth, we have in our microbiology fraternity, we had a huge discussion on this. But unfortunately, there was no conclusion that the only fact, or let us say a thought which was put forward, was that if by chance some organisms from the atmosphere getting carried with the raindrops,
Mhm. Yeah, that makes
and
sense.
they are specific for that fermentation, so maybe the outcome is formation of curd, was the idea that came forth. Otherwise, I don't think there's much of research which has been conducted on this
Okay.
yet.
He has another question. Bakery slash dairy products have probiotic bacteria, but on the other side, doctors tell us to avoid such foods. So how to have a balance between?
No, see, bakery products, if you are referring to cakes and breads, they cannot harbor the bacteria because they are baked at such high temperatures. No bacteria would
Okay.
survive that temperature.
Okay.
So there's no chance that if you eat bakery products, they would enter inside. And do some good. In fact, the contents of these bakery products are harmful to gut health.
Yeah.
And the dairy products, as you say, if you have pasteurized dairy products, it is written on the package whether they are pasteurized. So if you have pasteurized dairy products, it won't contain any of the organisms, because the process of pasteurization is intended to kill the organisms.
Right.
But on the other hand, you have what they do is some of the dairy products, especially yogurts and curds, if you see on their packets, after pasteurization, they add the specified good bacteria to it. But that is with very less number of dairy products. So when you pick up a pack, just check on that whether they have mentioned the addition of especially the lactic acid bacteria, as we call them, the good bacteria. So if there is a mention of those organisms on the product, then they are there. Otherwise, the pasteurized dairy products will not contain them. So it is always advised that the homemade curd, for which you established a inoculum, म्हणजे ज्याला मराठीत आपण विरजण म्हणतो, आणि आपण तेच विरजण दरवेळी वापरून तो फ्लोरा इनफॅक्ट आपल्या बॉडीमध्ये स्टॅबिलाइज करत असतो. So those type of products which have live bacteria would help.
Okay.
Otherwise,
Ma'am, do
then
you
have to
recommend? I'm sorry.
Otherwise, you have to depend on the probiotic formulations or the tablets that you get in the market. You do get probiotic tablets, which are actually doctors prescribe it after the antibiotic therapy and some such medications, which is likely to kill the normal flora of the body. So but they prescribe you on these medicines for a few days so that that flora gets re-established in the body. Or if you have observed ever that whenever you are going to have dose of antibiotics, normally the doctors also tell you, of course most of us ignore that, consume buttermilk or consume bread with that. Sorry, not bread, consume buttermilk or curd with that, so that it helps to maintain the flora in the body. But
Right.
most of us tend to ignore that and the flora gets disturbed. And that's the main reason why we experience bowel disturbances after antibiotic treatment.
He also wants to know whether we can get a link for this lecture. So, Dr. Samir, we will be uploading the recording of this webinar on our YouTube channel. So you can go check it out at jungle_bells. That is our YouTube channel. Also, we have live streamed this on Facebook. So you can go watch it on our page. Again, jungle_bells is our handle. You can go watch it on Facebook at any time. So one more last question, Dr. Pragati, is why, how you got interested in this subject, like how microbiology interested you, like how you got into it.
That was a very long time back. When I passed my 12th, I, of course, I had my inclination towards biology. And that time, the available option after medicine was microbiology. So I entered it. But from day one, the field interested me, and I pursued that. And it's a growing science, I would say, that every day you come up with something different which is related to microorganisms. So we have a lot of things to tingle the brain every now and then. And still very happy to share with you that still the subject as such, what I have been observing, now I am almost in this field teaching for 30 years now. So since I always had good response from students, but still I have students approaching that we want to do this only, madam. And after the pandemic, it has gone up too much that we want to pursue microbiology. And with so many good institutes in Pune, we have a lot of scope for the subject. National Institute of Virology, we have NCL, we have ISER, we have NARI. So there's a huge thing. I mean, my students have been doing well. Very proud to say that so many of my students are working with Serum Institute of India, which has got us the vaccine in form of Covishield. So that's very good, in fact.
Yes, absolutely. The interest level in this whole, all these topics has definitely gone up. And that was one of our reasons behind scheduling, this webinar, because, you get to learn a lot from experts like you,
Right.
which, otherwise from Google, that information may not be always accurate.
Because we tend to ignore the question mark which is given at the end of the sentence of Google, and we just read that part and end up with that.
Right.
So,
Okay. So thank you
so much.
Pragati ma'am. Thank you very much for being on our platform. And I would like to thank Sanjay Deshpande of Sanjeevani Developers, who is a mentor behind our venture Jungle Bells. He always supports us and helps us in all our ventures. And also Anuj Khare, who actually suggested your name, ma'am, to us and said that this can be a very interesting topic to have. So thank you, Anuj. And thank you to all our viewers for being on the platform and listening to us and attending our webinars regularly. Thank you so much. And everybody have a great day.
There is a chat box. It's mentioned 2 experts from NIV and Siagar College have joined Dr. Madhuri and NIV Dr. Kalpana of Siagar College of Engineering. So they have joined the lecture of yours today to listen you out.
Thank you so much.
Yeah. Yes.
All right. Thank you. Thank you, viewers, and have a great day.
Yeah, thank you so much.